Screening for genetic disorders. We talked about genetic disorders and we talked about how some of them could be recessive. So, for example, if the parents are carriers of the allele for the disease, they can screen the embryo in order to check for that and make the choice of whether they want to have the baby if the baby has the disease or if they do not want to have the baby. We have two general ways of screening embryos. The first is amniocentesis, and the second is chorionic villus sampling. Now, what is amniocentesis? Amniocentesis is a procedure that is performed around week 15 or 16 of pregnancy. It is performed by taking some of the fluid that is around the developing fetus. In the mother's womb, the fetus is surrounded by a fluid that is called the amniotic fluid. Amniocentesis is the procedure of taking some of this fluid around 15 or 16 weeks of pregnancy. This is because this fluid contains cells from the fetus. These cells can be used for genetic screening. We will talk about genetic screening later. As for chorionic villus sampling, it is performed earlier, between weeks 10 and 12 of the pregnancy. Instead of this procedure being taking some fluid samples, this procedure is actually taking a solid tissue sample from the placenta. The placenta is tissue that connects the fetus to the mother. So taking a sample of this placenta provides cells from the fetus for genetic screening. However, these two procedures are a bit risky. They can cause a miscarriage. And currently, there are techniques that are being developed in order to take fetal cells neither from the amniotic fluid nor from the placenta, but actually from the blood of the mother, in order to avoid coming in contact with the fetus and causing a miscarriage. So this is amniocentesis. A needle is injected through the abdomen of the mother into the amniotic fluid to take a sample of it. As for chorionic villus sampling, there are two ways to do it, either through the abdomen of the mother, insertion of a needle here to take some cells from the placenta, or it can be done by inserting a catheter through the cervix of the mother to take cells from the placenta. So the first method is the trans-abdominal method, which means through the abdomen, and the second method is the trans-cervical method, which means through the cervix. Another method involves the usage of IVF, in vitro fertilization. In this method, embryos are produced by IVF, so the parents decide to have an IVF. And then the embryos are tested to see whether they are healthy or affected. If they are healthy, they can be implanted into the mother to have a healthy child. And if they are not healthy, then the parents can try again. So these are methods of obtaining cells of the fetus, or in this case, the embryo, in order to perform genetic screening. So what is genetic screening? Genetic screening is simple. It is basically one principle. DNA from the fetal cells or from the embryo is taken and isolated and tested to screen the genome. That's why it's called genetic screening. You screen the genome to find the specific gene for this disease and to see what allele is found in the fetus or in the embryo. This is how you test for specific disorders in the fetus or in the embryo. If the screening shows that the child is going to be affected, then the parents have a choice here. Either they keep the child and they know that they will be raising an affected child with this disease, or they decide to have an abortion. If they do decide to have an abortion, they can then try again until they have a healthy baby. Because as we said earlier, if the parents are both carriers, then they have chances of having healthy babies and they have chances of having affected babies. So if they decide to have an abortion, they have a chance to try again until they get a healthy baby. They may choose to have pre-implantation embryo screening the second time they try using IVF to avoid having another affected pregnancy. So, if they have an affected pregnancy the first time and they decide to have an abortion, the second time they might go with an IVF to choose the healthy embryos.